And we have begun. So our endeavor over the next, hopefully just 3 periods, will be to get all of this in. A couple of things. I had a couple of students say they had trouble with some of those, that three different recordings. If you have trouble with, I can reset them and resend them. I always think sometimes you have to download them. You can always delete them and download them again. And if there's any persistent problem, let me know. I will resend them. I haven't really changed the exam, so everything is on there. Any consequence. So that's the first order. The second order of business pertinent to the email I just sent everybody. I don't even know how many people are going to be here on Friday afternoon to take this exam, but I'm guessing the number is going to be small. There'll be a few, but your neighbor this time. Boy, I'm telling you, the two of you. I'm going to separate the two of you. You and Brooke together. I know. Where's Allie? Taking a day off again? Okay, moving on. So I'm going to have a full class here of 28 or 27, 28 people, how many ever are taking the exam, and which means that I need, realistically, we'll have to stagger it. So to come in here, right, you have to be racing in between 2 rooms and setting it up in the uncomfortable confines of the lab is actually better. Statistically, take the exam where you have the lecture. So I'm going to ask those few folks to come in starting around 2 o'clock. It'll be easier. It'll get done. There's a fair amount of questions, but you'll get through them. I'm trying to remember there's 70 or 75 questions. So some people will just race through it and be done with it inevitably. Whether I applaud that or not is inconsequential. So without further ado, male and female reproductive. I am guessing that you had a lot of the basic anatomy. Oh, thank goodness. So we don't have to go through all the parts of the anatomy. We did this. Oh, yeah. This is slam dunk.
We went like really to depth. Yeah, you really want to.
Well, because it's
he
okay.
Glossed over urinary. Yeah.
Urinary is a big part of this exam, by the way. So urinary plays a role in the exam, I think. To be clear, I think respiratory is relatively de-emphasized. Probably less than 5 to 10 questions in respiratory, a whole lot more about urinary and male and female reproductive. So he didn't, then we're golden. I could take the rest of the day off. Maybe I should. Maybe I will. I'm going to use the Marie PowerPoints for 2 reasons. 1, this is really where the source of a lot of the questions are. And 2, for your viewing. So I'm going to buzz through these and stop me, please, if it's something you're unfamiliar with. And 2, because I have to, I'll get the other PowerPoint from the Welsh textbook for you. I just didn't load it. There's a lot of stuff going on around my place today. Basics. Effectively, this is another one of those systems where you have the primary organs, testes, and ovaries. Everything else is an accessory structure. Everything else. So the question, what's an accessory structure? Everything including, but not the ovaries and the testes, period. That's it. I'm not going to get into gametes and stuff like that, or this terminology, or zygotes, or gestation, or parturition. Important terms to know. You'll be, because particularly in nursing and for charting, we use like gestation and parturition as terms, because a lot of times it's how many times somebody gives birth versus how many times that they're pregnant. So it's grava and para, are the terms classically in Latin. You don't have to know it for me, but hey, I'm trying to help you. When I do the anatomy part of this, I typically do a lot of the homologous structures. I'm going to look through the test and see if there's any of this in there. But there's so many. Everybody, if you see the slides, everything looks identical. There really is, other than some. See, if we look at the starting points about them, there's not a lot of difference. We all start female for the first 5 weeks anyway. If you didn't know that, okay. So understand, you see a lot of structural commonalities. These are obvious. If you're going to look at the vas deferens, they're the homologous or analogous structures to the fallopian tubes. There really isn't one for the uterus per se. Those kinds of things that are there. Okay, we have the copulatory organs, the vagina and the penis. The clitoris and the penis certainly are homologous structures or analogous structures. You're going to have to know a bit about the testosterone, estrogen, and progesterone, all the different things. I mean, it's pretty straightforward what the sex hormones do once we reach puberty. I mean, they regulate the cycles when we talk about female reproductive. And in the male, basically, they're just part of all of these other things, whether it's sex drive, whether it's sexual behavior, whether it's secondary sexual characteristics, all of those kinds of things are pretty straightforward. Did he emphasize this, the axis? No, a little bit. Okay. This is critical to understand. Okay, this axis, basically, it's interesting that in female repro, we see a lot of positive feedback vis-a-vis when ovulation occurs. We see positive feedback with the onset of puberty in the male and the female, but it's interesting, particularly in the female. But, and we certainly see it with regard to the birth process. This is mostly negative feedback, and other than with the onset of puberty, there's a few little differences that are part of it. It involves this axis. I mean, a bigger part of this axis is really regulated by the presence of testosterone, estrogen, and progesterone. So effectively, the trigger, if those levels start to drop, then they're red, and remember the hypothalamus is the beauty of having a blood-brain barrier that's weaker. And what it does is, part of it is the posterior pituitary, and it causes it to secrete this gonadotropin. So it's just what it makes the gonads grow or be active. Release hormones. You remember back from endocrine, make things go, inhibitory hormones, make them stop. What that does is it triggers the anterior pituitary to make FSH and LH. FSH is follicle stimulating, and LH is luteinizing hormone. Not going to do all that, right? And the follicle stimulating is just follicles, eggs, sperm. Luteinizing, all of that is testosterone, estrogen, progesterone. Effectively, that's what they do. So their impact points are here, or here, and as these levels begin to increase, they negatively feedback, and that's the axis. So we say about the axis, those are the influencing factors, this hypothalamic pituitary gonadal axis. The other one is called inhibin, and inhibin is another one of these things that will feedback and very much inhibit the release of that. So that's a byproduct of production where there's sufficient counts of, particularly of sperm, I would think, as much as anything else. So you can take a look at that and read through it as much as you want. Okay, gonadotropin release hormone, the FSH and the LH from the anterior pituitary, sex hormones that are there, negative feedback, inhibin, gonads of both male and female. They also exert a negative feedback, particularly on follicle stimulating hormone, because it's really, as long as there's satisfactory egg and sperm production. I think sperm production predominates, but I'm not going to ask you about that. Okay. The way it works. Really important access. Very good graphics, or look at it. And I'm sure you use similar graphics or different graphics for your stuff. Similar, different, typically a combination. So puberty. Okay, when reproductive organs adult size and become functional, theoretically the earliest time. Unlikely at the onset of puberty with the first start of menstruation or the first part of ovulation, or for that matter, the first time the male could accomplish ejaculation, that you're going to necessarily see fertilization, more so on the female side than on the male side. Stein's, how should I say it, disclaimer. This topic is not my long suit. I've told you before, every professor has stuff that they're good with, stuff that they're not with. My context for this is from the concept of infertility. I know I'm very good at knowing what goes wrong. I've spent a lot of time dealing with that over the years. So that's my understanding about what goes right. Is sometimes it's miraculous. It amazes me. So before puberty, very low levels, all they need to suppress it. As puberty approaches, for whatever reason, it becomes less sensitive to those levels of estrogen and progesterone and testosterone, and all of a sudden, boom, it just goes crazy. Why it happens then? There's a lot of theories behind it. It may have to do with stuff in our diet, which has changed significantly. A lot of the hormones we're putting in feed and things. I mean, you can go. It has impacted the earlier onset of puberty, and interestingly, it has impacted a greater amount of problems conceiving. I mean, it's not supposed to happen that often. My wife and I could not. We're considerably older, but my son and my stepson, each, my eldest two kids are IVF. My stepson's two kids, they're one kid IVF. She's pregnant now with number two. Well, what does that tell you? I mean, it's a pretty small subset for obviously no biological ties for me from that. Okay, and so the anterior pituitary then releases the FSH and LH, stimulates them. The threshold of inhibition continues to rise. In other words, usually rather than inhibiting that threshold, it's still negative feedback, but it just makes it harder to negative feedback, if you want to think of it that way. And they're released until the adult pattern is finally achieved. So that's what happens. So puberty is relatively profound and proceeds relatively rapidly at the onset. I am not doing it. I don't think I have any questions on meiosis and any of that stuff. I'll double check. I'll even check tonight if you're nice. Okay, I'm not doing sister chromatids or homologous pairs. This has to do with the whole idea of meiosis is one thing, diversity. The whole idea of mitosis is identical copies each and every time. There's two major things that occur in meiosis that are different. You should know these going forward. I don't think there's anything on them, but this is what makes the diversity diverse. And we remember there's two phases. There's meiosis 1 and meiosis 2. In prophase, what we call prophase 1, prophase of meiosis 1, there are they form a tetrad. You have the sister chromatid double and sit sister chromatids here from mom, sister chromatids from there. They form this thing they call a tetrad. They're in very close proximity, and what happens is the ones that are this, they cross over. There's a lot of terms for it. Synapses, crossing over, and effectively what happens is little bits of DNA from each chromosome kind of swap places. So eventually when the mom's side and the dad's side, we have variation inherited. So that's the first part. We see that in prophase 1. The second part occurs just after that in metaphase 1. Metaphase 1, remember what they do. They line up, right? You have that equatorial plate type of thing. And all portions, so to speak, line up and then they're pulled apart. Remember that a little bit? It's called the law of independent assortment. Dude, there's 23. There's a lot of assortment that goes on. It's not like, DNA chromosomes line up, starting at number 1 all the way to number 23. Everybody facing forward. Mom chromosomes do the same thing. It doesn't work that way. I mean, 1 isn't followed by 2 necessarily. 2 isn't followed by 3. 1 could be forward, 2 could be backward. 1 and 2 could be forward. 1 and 6 could be forward. 3 and 7 could be backward. And I have no idea. So there's all that independence of 23 pairs lining up either forward or backward. So you don't know where the next chromosome is coming from, so to speak. Those are the two major things for diversity, and we're not even including mutations in the normal course of events. Effectively, mutations that always occur, as we have discussed previously. We're a little bit in AP1. It's a living system. Stuff happens. So there's the crossing over, and once Again, they're haploid. Once they come together as diploid terms, you should know. Again, if you haven't had it by this time, I'm surprised. And they form the zygote. Don't care. We're not reviewing it. Same names. PMAT. Remember that from AP1. Prophase, metaphase, anaphase, and telophase. Okay. Here's the reduction. Nice, pretty pictures. Look. Mitosis does this. Meiosis does that. Genetic diversity. All daughter cells genetically different. Random alignment of the pairs. That's that independent assortment. I didn't discover that. Some monk from Belgium who happened to be living in Prague, Czechoslovakia, did, named Gregor Mendel. He didn't have a computer. He didn't even have a microscope. He just had some plants. Remember Mendel, all that thing, pea plants, pea pods. Pretty smart guy for back then. He's a monk. What else did he have to do? I'm counting plants. Sorry. Bad humor. Does Allie only come every other day? Yeah. You're not our keeper. Not doing that. Give me strength, Lord, from this. Not doing any bioflex. All right, we're finally here. So you did all this. So once the sperm are produced, I'm not going into all that stuff that are there. Basically, what happens? Key factors there. Obviously, they're produced. Where are they produced? The seminiferous tubules. You got that before from Dr. Robbins. Okay, I'm not going to go crazy. The testosterone is produced in the same area, but it's called a different kind of a cell. They're called the interstitial cells of Leydig. There's supportive cells that are there. They're a little myoid, which means muscle-like cell, that kind of get the sperm moving on their journey. Where they go to after that, not directly, but the series of tunnels is called the epididymis. That's where, at that point, they're almost just like typical tiny cells, circular, if you will. They're going to form the head, the tail, the motor area, the midpiece in the middle, the acrosome, the sort of helmet-like structures on top. That's where they mature, and they eventually gain the capability of motility, of being able to move. Critical piece. And they are stored there for a rather long time. And effectively what happens at the time of ejaculation, they are pushed out of there. That's where they are stored and pushed out on their journey. And it's a relatively long journey in the male, by way of what's called the VAS, VAS, or ductus deferens. Either term works. Basically, since the urethra, you got that from anatomy, since it's a shear system, okay, they merge with the urethra, okay, eventually within the prostate. Prior to that, they merge with something called a seminal gland, which produces a lot of the fluid, okay, and that's in the ejaculatory duct. That ejaculatory duct meets the urethra in the confines of the prostate, and then on its way out from there by way of the urethra. The bulk of the fluid that's part of what we call semen, getting its name from the seminal glands, is from the seminal gland itself. About 2/3 of it, 70%. There's about 30% from the prostate. The bulbourethral glands are just lubricant. They don't add to the ejaculant per se. So and that's a very unique system because there are so untold millions of sperm. We're talking about an ejaculation, which is maybe between a half and a teaspoon of fluid totally. Maybe we're talking a couple of 100 million sperm. You need a whole bunch. Half of them go the wrong way. I didn't know. I thought it was on the right. When it comes to a fork in the road, take it. I hate to be flip about it, but we got to have fun. If you haven't got me fired yet, this is your chance. I see you taking notes. Just writing it down. Moving on. So the idea, you have this vast number, so many of them are drain out, So many of them are clobbered by immunological reactions. So very, very few of them make their way. So if you think of it that way, just think how tiny they must be. Part of the things they look at for infertility are sperm count and effectively how modal, how moving these little entities are, so to speak. That's part of it. I'm sure you saw this. It's a very long system. What they have in common analogously is that white. Both the ovaries and the testes have two tunics. They're surrounded by peritoneum. Because the testicles descend, this is peritoneal tissue. He talks about the descent of the testicles, correct? Okay. In fact, why is that accomplished? I mean, the why is something we can all subject to interpretation, but effectively, functionally, what they do is they reduce the temperature. It is cooler, and the optimal temperature for sperm production is about 2 to 3 degrees Celsius less than normal body temperature in the interior. The eggs, it doesn't matter. Okay. And so there's this very interesting structure called the spermatic cord, which has what? Lymphatics, nerves, a ton of blood vessels, because that absorbs the heat. Very elaborate venous plexus to absorb the heat. And it makes its way down through a weakness in what we call an opening, a tunnel in the inguinal ligament. That's where sports hernias or ruptures occur. And they descend. And effectively, and that's why they're so sensitive, you have peritoneal tissue, a ton of blood vessels, nerves, lymphatics. And, if somebody's struck there, if you see it, in a football game, in a soccer match, somebody blocks a soccer, big problem. So that's part of what happens. And so that's one of the reasons, one of the first things they look at in male infertility is to see if it's a temperature thing. So if you're having difficulty conceiving, it's the difference between using boxers, which will bring up the temperature because they bring the testes up, versus briefs. I mean, the other way, briefs bring them up, boxers. One of the first things they tell you if you're in infertility counseling. There's a lot of things that are about that. I mean, I won't be able to share everything with you in the time we have. But here, they're stored in the epididymis. They mature. And then this is a rather long journey all the way down on either side of the bladder, obviously. And now you can hear the large seminal gland as they come together. This becomes the ejaculatory duct. So the ejaculant then forms. And then, again, they join the urethra on its exit point from the bladder within the confines of the prostate, which is about the size of a peach pit. It's not really terribly large. And then effectively that's called, now it's the prostatic urethra. Then you have something there called the membranous urethra, and the elongated portion around the erectile bodies, the corpora cavernosa times 2, and the corpora spongiosa is the urethra. So all that's part of it. So I'm sure this should have all been covered in the anatomy, correct? Got it? Good. Happy days. So here we are outside, 3 degrees Celsius lower, sperm production. Yeah, it's divided. The names of the two muscles anatomically and I never forget which is which. They're the ones that in cold temperatures will pull up, those types of things where effectively they're structural change, not structural, but positional changes that occur in cold weather after getting out of a shower or something like that. The dartos and cremaster muscles. I'm sparing you the bad jokes. No Seinfeld fans here. Too bad. They did a whole thing on that. It's a show about nothing. This is all the vasculature. One of them is the cremaster, one of them is dartos. I don't remember, not going to ask you. Oh, yeah. In the male, we talk about the prepuce or foreskin, which in this country we don't see a lot because it's typically removed surgically. Plays a very important role microbiologically. Much more people are much tears in the male, and it's much more predisposed. It's a really high risk factor for HIV/AIDS. Did not know that. So a lot of them are done for health reasons. I mean, it's religious reasons are certainly part of it. They didn't give me a choice. 8 days, boom, going. Judaism, that's the whole covenant thing. Just saying. Thanks a lot. Next time, give me a choice. These are the two things that that both the testes and the ovary have. They have an albuginea, that's that white outer capsule. It's very strong. And the vaginalis, which is derived from the peritoneum, that's the soft exterior lining. And here you have the divisions. You don't have to know this for me. Okay. Again, the venous plexus, what they do, you don't have to remember the name. The spermatic cord, you should remember the name, all the stuff that's in there. Okay. Here you go. Can I have at least one bad joke? Thumbs up. Thank you. I see this one thing. Ramen noodles. Not bad, was it? It was worth a giggle. College students and ramen noodles. Brooke's finally getting it. Doesn't remind you of ramen noodles? But it's really one continuous set of tubes. All of these tubes are just more tubes. So think of how, again, it's this repetitive concept of having a lot of surface area, whether it's just a series of tubes that are, all, and eventually all the sperm that can be stored in there before they are, and effectively they're stored all the way up here from the head, all the way to the tail, and then ejaculated that way. And then that's the start of the Vassar Stephen's illustration. Cryptorchism, undescended testicle, more common than you think. Okay, extraordinarily strong association is with risk factor for cancer. Testicular cancer, no joke here, everybody. 15 to 35 was common cancer in men. Thankfully, very, very treatable. Whatever you might think of Lance Armstrong, sort of a disgraced, unbelievably great rider, he overcame that testicular cancer that had metastasized to his brain. The very fact that he did that and could win like 7 Tour de France, legally or illegal, enough speaks to his athletic ability. And interestingly, cancers are more often painless masses than painful masses. Mario Lemieux, when they discovered the mass here on the back of his neck, which turned out to be Hodgkin's disease, I mean, he's talking to the trainer. He goes, "Yeah, I got this bump on my neck, but it doesn't hurt. Forget about it." And the trainer, on top of his game, goes, "Yeah." Feel it around. He says, "Doesn't hurt you at all. Pretty solid mass. Let me get team doc in." Next thing you know, and he had a family history of Hodgkin's disease. Just, you don't want to never be dismissive of things, the moral of the story. So there's a lot of cures with regard to that. The back part, and I touch on this a little bit in my micro classes, men are very reluctant to do self-testicular exams, or they should. Self-breast exams are imperative. Women, as far as I know, are less reluctant. At least I have been told. Okay? Again, the watchword is women have babies, men don't. Men are much less. I'm going to say it again. Forgive me. As long as the system works, guys are fine. Don't get too excited about anything. And only when something bad happens do they get to go to the doctor. Here you are, 20 feet stored in there in the epididymis, non-motile sperm, 20 days roughly, stored for several months, etc. You can see the ductus or vas deferens relatively long. Okay. So I mean, that's about 1.5 feet in total length, ballpark. Vasectomies, very effective can be reversed if you did not know that. And I mean, that's the most effective means of that. And when I say it can be reversed, my wife has 5 kids. The 3 before were pre-vasectomy on her ex. Post-vasectomy are 2 more. So yeah, very reverse. The 3 parts, that's anatomy, prostatic. You should at least know prostatic part and the spongy part. That's the most significant part, because that's really when we're starting to talk about the physiology of the intermediate parts, just going from inside to outside. Again, the erectile bodies here, the cavernosa, the large ones, paired on either side. As you can see, the urethra dwells centrally So even though you have the erectile bodies, the circular nature, what it does, it doesn't squeeze shut. That would be counterproductive. It maintains the opening. And there are means to prevent the urine from joining the ejaculate. And all that type of, here's the circumcision nugget, the relationship to HIV. Okay, here are the erectile bodies ending in the bulb that are there. They whatever physiologically, whatever the stimulus. The male is relatively complicated, is more complicated anatomically, very uncomplicated physiologically. Totally different. Female is less complicated anatomically, much more complicated physiology. And again, we have to go in to all of the, the different phases and the names. It's a little confusing. So from a test taking, if it says in the uterine phase or versus in the menstrual phase or whatever, immediately that focuses you on what you have to know. There's one illustration in this PowerPoint that makes life easy, I think. And the perineum, you just have to know what's in there anatomically, basically between the pubic symphysis anteriorly, the ischial tuberosities on either side, and the coccyx posterior. And there they are. And if we see the illustration for the female, which we won't get to today, it makes you understand how transitional changes are possible and effective, whatever your feelings are about those. So what does the seminal gland do? 70%. Everything here that controls, just like digestive, are pH. Everything is about pH here. So this is an alkaline fluid. Its principal role is to neutralize the lactic acid. If you didn't in your micro, you should be talking about the role of lactic acid, which is the principal defense against infection that's in the female reproductive area. Something called lactobacillus, because of an abundance of estrogen, making an abundance of glycogen, which makes lactic acid. Okay? You have to coagulating enzymes to prevent leakage out. The only energy source is fructose. They don't run on glucose, they run on fructose. And it's a viscous fluid. Prostaglandins are the local mediators. They got their name from the prostate gland, even though they're all over the body. And this is the part when you hear about on crime shows, when we use an ultraviolet examining light a lot in medicine. They use it a lot in crime scenes. You can use it in crime scenes with something called luminol to detect if there's blood that is still there or traces of it. And this is that one that they lose for fluids to show whether it's a rape scene or a sexual assault or any kinds of fluids of this nature. Just what was going on in that room before something bad happened. Okay, that's what they're using. Very easy to see because of the yellow pigment that fluoresces with ultraviolet light. When I talked about my class of microwave dermatology, we always had basically the same light that they use, called the Woods light. It's just an ultraviolet examining light. It's not ultraviolet that's dangerous to you. So the prostate, peach pit, this is the milky white part that's characteristic of the fluid that most people would recognize. That is there, the color of it. This is a big deal. It also has what's called PSA, prostatic specific antigen. This is just like breast cancer. The most common glandular cancer in men is prostate cancer. The way that we analyze, there's two tests that are done after a certain age or in the presence of a certain history. Let's say over age 40. In men over age 40, they do a digital rectal exam. Okay, and to palpate, from the rectum, you can palpate whether the prostate is soft or enlarged, or whether it's rough or hard, all of these things. The other thing is called the PSA. It's a blood test. Those are the two criteria to be used: presence or absence of cancer. If there's anything suspicious, you might have to do a biopsy, but that's a different story. Presence or absence of cancer, and if there is cancer, how well your treatment is succeeding or failing. If the PSA goes up, not good. If the PSA starts to go down, stays down, or gets lower, then you're monitoring treatment. So every year I have a physical, every 2 years when I have a physical, they do this routinely. Even though, if you did not know, October of 21, I think I had the bulk of my prostate removed, not for cancer, because it was so enlarged from all the treatments for my bladder cancer. So it was not, thankfully, it was not a cancerous thing, but just to give you an idea, they play a role, non-specific, in activating the sperm. Again, about a third of the body. So it's mostly fluid. These don't do much. Lubricant, there's Bartholin glands in the female that kind of do the same thing. And as you can see, there's a tiny, tiny amount of acidity. That's not enough to neutralize. So the acidity is just the nature of the fluid. It doesn't offset the alkalinity of the fluid from the seminal gland. Okay, here we are. After half men after 50, 80% of men after age 70, it's pre-SOP. It's a lot of times fairly easy to treat. Most of the time, if it's not a problem, there's a medicine, whose generic name escapes me, called Flomax, which works very well, relaxing. And years ago, the meds had a lot of side effects. Okay, and all of these things are part of it. Medications. I had a laser procedure done. Procedure was easy, didn't bother me. The healing part was very unpleasant. And prostatitis is a bacterial, I cover this in micro, is a bacterial infection, sometimes associated with prostate disease, but it's not something that can be spread. To our knowledge, it's not able to transmit to a partner. And here, big number. If it's a cause of death, it's very slow. If someone dies of prostate disease, it was either untreated or undertreated. And it is typically, if someone is treating it, it takes a long time. It's not like lung cancer or stomach cancer or pancreatic cancer. No. People die of prostate disease generally, again, men, they sometimes ignore things. Here's the semen, 20 to 150 million sperm, fructose. Alkaline fluid neutralizes the acidity of the male urethra and female vagina, enhances motility. That again, pH control, prostaglandin. And this is the brilliance of it. These local messengers, what do? Decrease. There's a mucus that's in there that allows sperm to pass. Relaxin, that basically relaxes the surrounding structures again, and enhance sperm motility. ATP for energy. Okay. Suppressing, there's an immunological response. We used to call it hostile mucus. That's something when you do an infertility workup, you look for. There's a test my wife and I did after showering, first thing in the morning, you have intercourse, and very, very quickly go to the doctor and they examine the sperm and the surrounding fluid to check sperm counts and things like that. It's called a PK, or postcoital, even though they're spelling it wrong. We had this workup done back in 1980 by at the time, the man considered the world's leading authority on infertility at Hospital University of Pennsylvania. Unfortunately, it was unsuccessful for us. Friends of ours, their consult there was successful, so they treated her for endometriosis. Unfortunately, she's no longer with us. So our kids were born very close to each other. Hers biologically, ours obtained via adoption. Now being a menace in the Pentagon, as you know. Be there, Grace. She's asleep. I'm not looking. Love her to death. Clotting factors to prevent it from draining out. Okay. Yeah, so all of this coagulates the semen to prevent it from draining, and then it's miraculous as far as I can tell. The physiology, not much to it. Again, parasympathetic is the erotic aspect of it, the stimulation. Okay, all of that types of thing that are there, and it's by way of the parasympathetic. There's a zillion things that can do it. Okay, and the sympathetic. So we're talking about the arousal period in the male or the female, parasympathetic. The climax or orgasm or ejaculation portion of it, be it male or female, that is sympathetic nervous system. We talked about that in AP1, in autonomic nervous system. the mediator of this, the parasympathetics release nitric oxide. Nitric oxide, its main job is to dilate blood vessels because of the structure of those cavernosa that are there. So the parasympathetics that are triggered are the ones going, okay, into the reproductive areas, and they cause engorgement or enlargement because there's so much nitric oxide filling that effectively these veins almost get overfilled, and effectively they're so packed so tightly the exit ways are no longer available. So the better way to say it, retards venous drainage. After that dilation, the spongiosum even is part of this, but it keeps the urethra open. All sorts of things can be inhibited by all sorts of things there. And these are all things when you study infertility that are part of it. And a lot of these are sexual dysfunctions. We learn about those things that, play a role in it. And then they go through the reflex that's involved here. And you don't have to go crazy with that. The release, a relatively rapid expulsion, that's there, and that's the climax or orgasm in the male. We're almost there. Hold on. Okay. Oh. Sorry, kind of important. And so this is, and effectively, well, not so in the female. In the male, there is a refractory period, varies with time, varies with age. Again, there's a lot, and effectively the resolution is when all of these things kind of revert back, and before they can be re-triggered again, there's a certain amount of time that's involved. Okay. So this, and then we hear a lot about these. Most of these, as you can see, is a rather large. Why we have all these problems as well, on top of the infertility, is something of significance. Obviously, there's other factors. Alcohol plays a role. Psychological factors play a role. Things are happening in your life. All of those things. Physical condition play a role. But the reality is that at least these can be treated, and by and large, all of the agents work on, it should be a capital O, nitric oxide. And what they do is they prevent it from being broken down. For all medicines, there's a metabolic pathway that makes them effectively breaking down so we can excrete them. This just holds it in longer. So it prevents the nitric oxide from being broken down. So it's a nitric oxide, I'm trying to think of the right way to express it. So it just prolongs the activity, would be the best way to say it. What's interesting in the male, and these are just some of the cells I told you about, we could skip through mitosis and type A and type B, not doing it. Parts of it, the head, the acrosome has the enzymes to be able to penetrate the egg. It has the genetic material. The mid piece is just for ATP production. The tail is for locomotion. The head is where it all counts, and the acrosome just allows it to enter. Good illustration for it. Very important, the blood testes barrier that's there. As you can see, sperm are produced at puberty. Our immune system doesn't recognize them. They recognize them as foreign because they were not there as birth. So if there is an accident, or maybe a surgery that went wrong, or some kind of infection that caused release of the sperm into the bloodstream, your immune system would attack them as invaders. So this is one of those, like the blood-brain barrier, there's a blood-testicular barrier, okay? And because of that, that's something you keep in the back of your mind in cases of trauma to this area. Again, I'm not really that concerned about that. One in 7 couples, decline in fertility, all these things are part of it. All these, you can read through these, there's a lot. It's involved with it. Here's the axis again, not going to go over it. And what's interesting is this chart. And it's different with men and women significantly. Pre-birth, we can understand. This has to do with organ development. Why you have that little spike post-birth, I don't know. Maybe it's the finalization of organ development. I'm speculating. Maybe it's the finalization of the testicular descent functionality. Could be. And then it's gone from early during infancy until puberty. And once puberty, almost immediately, certainly, within a couple of years of the onset of puberty, it's at adult levels and is maintained there well into senior years, 70s, 80s. There's a lot of evidence of people with or without, not talking about in vitro or things like that, being able to father children with men who were in their 70s, 80s, 90s. Nothing unusual about that. Today, given the state of IVF, I think it would be comparatively easy to do if you did it indirectly rather than, let's say, in a traditional or more natural method, even if that's the right way. So you'd say about 3 years to achieve it, fairly stable throughout life. And again, if we didn't have this, there'd be big problems. I mean, all those types of things, if we didn't have that. We all know about the secondary sex characteristics. And we did male. Thank you. It was like a race, but I got it done. Female takes longer.